Good day and welcome, everyone. On behalf of Cambridge Healthtech Institute's Global Web Symposia Series and our sponsor, AroCell, I'd like to welcome you to improved monitoring of tumor proliferation with a novel serum biomarker, AroCell TK210 ELISA. My name is Elizabeth Lamb, and I'll be your host and the moderator for today's event. At this time, I would like to introduce our presenters for today. First is Stefan Eriksson, professor in medical and physiological chemistry at the Swedish University of Agricultural Sciences. And our second presenter is Martin Shaw, business development manager for AroCell. So with that, we will start with Dr. Eriksson.
Thank you very much for your introduction, and I will tell you today about a new serum biomarker called AroCell TK210, and it's in an ELISA format, and this one can be used for monitoring tumor cell proliferation. And the point of what are tumor markers is simply that any substance that is associated with the presence of a tumor can be used as a tumor biomarker. And most serum tumor biomarkers is indicative of the total tumor mass. Examples are CEA, PSA, which is most known usually, but also a whole series of other, including CA15-3, that we will describe a little bit more later on. However, there are very few serum biomarkers that are indicative of cell proliferation. Why is that important? Well, since most current biomarkers can be said to measure what have happened, and that is the past rate of change in the tumor disease, that is related then to the doubling time of the tumor. And therefore, the current situation is related to the total tumor mass. However, what really matters is the growth of the tumor. And therefore, a proliferation biomarker is very much needed because it may provide a measure of the tumor growth. It can be used to stratify patients in different groups, and it can aid in the choice of therapy, so that when a proliferation is low, there is a less aggressive therapy needed, and you can apply what is often called watchful waiting. However, if proliferation is high, you need a more aggressive therapy. So therapy is often drugs, chemotherapy, and many cancer drugs are toxic and have several side effects, some of them very severe. They are also expensive, and there is always a risk of therapy resistance. So if we return to what are the current proliferation biomarkers, well, they are so-called immunohistological biomarkers. They are measured in tissues with microscopy. And the ones that are available is called Ki67. It's another one called pCNA, and thirdly it's one called TK1. And those are all specific to cells that are in the so-called S phase, that is the synthesis phase, when a new DNA is made and when the cells are preparing to divide. These are important, but they have their limitations. And as I mentioned, these require biopsies. You have to make slides of tissue samples, and therefore they are invasive. And they only usually shows a small part of the tumor, not the whole tumor. Also, when you look into microscopy, the operator has a large influence, and there is often rather low reproducibility between different histological analysis. So there is a need for a blood biomarker, and that one then should be able to indicate the total activity of a malignant disease, and it is a very big advantage if it can be relying on a standard serum or blood sample, and that it is determined with a routine analytical method available in any clinical chemistry lab. Serum thymidine kinase 1, we call the TK210, has the potential to be such a marker. So it is already known as a tumor or a proliferation biomarker by using methods to measure its activity. And it's been used for over 35 years, but really in hematological malignancies, blood malignancies. It has been studied in many other types of solid tumor diseases, but not the activity assay, not clinically used today. It is also known that when you use it in immunohistology, the staining the reaction is very much associated with the gold standard marker Ki67, as I mentioned previously. So what is TK1? What is it doing? Well, it is released when the cells are growing in an abnormal way at high rate. The enzyme itself is involved in the phosphorylation of thymidine, one of the 4 building blocks for DNA, and ATP is used to transfer a phosphate to form thymidine monophosphate. This is the basis for all the activity assays, and there are different ways to measure this reaction. The TK1 is released into the bloodstream as part of a necrotic process. That is when the tumors first have grown and then they are disintegrating. And then TK1 comes out into the blood and appear as large complexes. And those complexes are both active and inactive in terms of enzyme activity. So how is thymidine kinase 1 used as a proliferation biomarker? Well, immunohistochemistry, that has been used quite a lot, have shown that in breast cancer, ovary cancer, cervical and prostate cancer, lung and kidney cancers, one can detect it, and it's also increased in case of cancer. In serum then, the enzyme activity measurements have shown very clearly that TK1 is increased in leukemias, in different forms of lymphomas, in myelomas, and so on. There is also a direct relation to therapy response, and one author has concluded that for patients with recurrent breast cancer, high tumor TK1 activity is a significant marker for poor clinical outcome in chemotherapy. So how does TK1 look in serum? What is the structure of TK1? Well, inside the normal cell or the growing cell, it is a tetramer, so it's 4 units of a subunit of, and however, in case of TK1 in serum, in the blood, there are large complexes of, or polymers of very different size and enzyme activities. And therefore the enzyme activity assay often can underestimate the total amount of serum TK. And an immunoassay, an assay based on antibodies, can solve this problem. So here is a model of TK1 in the cells with 4 units. And in the blood then, it's large stable aggregates of many of these tetramers, and in different forms. So some of them are active and some of them are much less active, but they all express the TK antigen. This is the antigen here in red to which the antibodies react that we have utilized in the assay. So currently we measure enzyme activity in the assays that is available on the market, but some of these aggregates, especially from solid tumors, are not enzymatically active, but they all express TK2/10 antigen. This has been shown quite clearly in an experiment where we separate the serum from blood donors and persons with prostate cancer in a molecular weight type of chromatography, where we collect different fractions, and in this case the high molecular weight, the larger complexes, come out in the first fractions. And in these fractions we measure TK activity, and you see from blood donors they migrate at high molecular weight. In case of prostate cancer patients, they are in multiple forms. And more specifically, if you look at the protein levels here in a so-called Western blot, where the antibodies has been used and you separate the proteins, you see that a certain fraction has no activity, but there's quite high levels of protein. So in subjects with solid tumors, much of TK1 protein exists as inactive TK1. So what about the assay? Well, my colleague Martin Shaw will soon tell you more about these type of assay, but I will just shortly here describe. It's a micro-titer plate, a so-called sandwich standard ELISA, using 2 monoclonal antibodies, one on the plate and one in the bottles here, and a unique pre-assay treatment, and we'll hear more about this later. So a little bit of the studies that have been performed, some results. And one important observation in recent years is that if you study a group of patients with a hematologic malignancy, blood disease, you can use so-called ROC analysis, a receiver operator curve. In this case, it's the sensitivity and specificity that is determined in this group between healthy and diseased persons, and you can see that there is an area under the curve that this value is particularly interesting. It shows the efficiency of the diagnostic method. In case then of hematologic malignancies, there is very little difference between the two methods. So the overall, both methods can be used for this type of cancer disease. However, in breast cancer patients, a group of patients, and when this is measured, you see that the concentration determination is much more efficient, a much higher area under the curve than the activity measurements. So there is about a 3 times higher sensitivity of the TK2/10 ELISA. This is even more useful when you combine the TK2/10 ELISA with another cancer biomarker, the CA15-3, which we mentioned earlier. If you measure a group of patients with breast cancer, you can see that each alone has rather high accuracy, high methodologic precision, but when you combine them, in this case, you get a clear increase and you obtain very high sensitivity and very high area under the curves. So in conclusion, the AroCell TK2/10 ELISA is a biomarker related to cell proliferation. It reflects tumor proliferation. It can be used to study many types of tumors, and it has a capacity to better discriminate between healthy controls and subjects with solid tumors. It therefore can aid in prognosis and therapy selection and in monitoring of a therapy. And it adds information when you combine it with other biomarkers. This is my last slide, and I hope we have shown you that this new assay is related to very important measures that really matters when you use a biomarker. And with this, I leave the word to my colleague. Thank you.
Good afternoon, ladies and gentlemen, or good morning, depending on where you are in the world. And thank you for taking the time to listen to Professor Eriksson and myself. I'm now going to present the TK210 ELISA, but this time more from the laboratory point of view. And my agenda will cover the following. First, a bit more about TK1. I'll repeat the points that are important from an analytical point of view, and then the assay procedure, performance data, sample stability, some further information, and then discussion. As Professor Eriksson said, TK1 is present in serum as stable aggregates of many molecular weights and enzyme activities, but it all express the TK21 antigen. And this is important because, as Professor Eriksson said, a lot of TK1 protein in serum is enzymatically inactive, so it's missed by current assays, but it expresses the TK210 antigen and so can be measured by immunoassay. This is the assay. It's a standard microtiter sandwich assay. It utilizes monoclonal antibodies raised against a unique immunoassay sequence, the TK210 sequence. And importantly, there's a pre-assay treatment buffer that releases the TK1 units and exposes the TK210 antigenic site. Here we have the method. First of all, you pre-treat the sample to release the TK1. You add it to a microtiter plate where it bounds. You then add a labeled antibody, labeled with biotin, and then you develop the color using horseradish peroxidase that has been labeled with streptavidin. A very standard microtiter procedure. It's for the assay of TK1 protein in human serum. It's got a wide assay range. It's sensitive. It gives linear assay dilution, so you can dilute samples. It's very precise, better than 5%, and it's very stable, so you can have it in your refrigerator waiting for samples to arrive. TK1 itself, the protein, is very stable. It will last a day at room temperature, so you can process the samples. It's stable at 4 degrees for 5 days, so it's easy to ship. And then at freezing temperatures, it's got long-term stability, so you can collect samples for future assay or do retrospective studies. Clinical information is now being developed, such as this article. Please contact us for more information. As Professor Eriksson said, in hematological malignancies, the data is very similar to that found with current enzyme activity assays. And this is useful because it means you have an information base to build on if you start studying these diseases. But also, looking at solid tumours, you have a much more sensitive assay due to the ability to detect the inactive forms of TK1 which are found in these tumours. And then, being a straightforward microtiter ELISA, it's easy to multiplex with other biomarkers to gain increased knowledge and information, such as here combined with CA15-3. So to summarise, the TK210 ELISA is a standard ELISA procedure unaffected by serine inhibitors. It's a unique sample preparation, so you measure all of TK210 and all of TK1. Good correlation with enzyme activity in hematological malignancies. Better discrimination between healthy controls and subjects with solid tumours. Easy multiplexing with other tumour biomarkers, and it's CE marked. So thank you, ladies and gentlemen, for listening to Professor Eriksson and myself, and I hope we convinced you that by measuring proliferation biomarkers, you're measuring what really matters. And I now throw the meeting open to discussion. Thank you very much.
Thank you very much, Martin and Stefan. We do have several questions here, and for our attendees, please feel free to chat those questions in using the chat panel. Our first question, what about the specificity of TK1? Are there other sources in the human body that express TK1? And if so, under what conditions? Inflammation, for example?
Okay, thank you for this question. It's a good one. So this is Staffan speaking. So I think it's clear that there are several different sources. Actually, all rapidly proliferating cells in the body will provide some TK into the blood. And it is also so that things like inflammation or an active form of infection will increase the TK, but only temporary. So that for being specific for tumors, it's really the stable high level of TK that is the biomarker for a tumor, and this is a very specific process that can be used clinically.
All right. Our next question, what about TK1 and other biomarkers in prostate cancer?
Okay, this is also a good question. Of course, in prostate cancer, the most used and most famous biomarker is the PSA, prostate-specific antigen. But I will say that TK is used now, and we are inquiring about how to use it in combination with PSA. But I will refer you, there is going to be a presentation at the Cancer Research meeting this fall. This will be in April, 2nd of April, and we have a poster number 715 that will be presented by our colleague Kiran Kumar. And so we have an abstract in the AroCell website, and I can also refer you to the list of ongoing clinical studies, including a very good study on prostate cancer, and this is listed in the AroCell website on the page Research and References. Please visit this one.
All right, thank you. We have a question from Lordanas. In the breast cancer data, which TK activity assay did you compare with? Liaison?
We do have some studies with Liaison, but in that study, this is Martin Shaw speaking, we compared with the gold standard tritium-labeled thymidine assay. So in our studies, we always refer to the gold standard tritiated assay, but we do have other data showing a good correlation with the Liaison assay.
All right. And a question from Ronit. Are there any users currently testing the assays in Europe?
That is so. There are several different sites, but I refer to the AroCell homepage also, because we are testing it here in Sweden at several places. So I think there you can get an overview.
All right. Another question, what clinical trials does AroCell currently have underway?
Well, this is Staffan again. I think I would like to visit this website, and we have several different both retrospective and prospective studies ongoing. So I think it will take a little too much time to list them here.
All right. How does the TK210 ELISA compare with enzyme activity assays, especially the Biomarker assay?
This is a bit of a follow-on. This is Martin Shaw, and this is a bit of a follow-on from the earlier question about Liaison. But one must remember that the DiviTum assay is an enzyme activity assay, such as Liaison was. And one must be careful that when you're comparing an enzyme activity assay and ELISA, you're comparing apples and pears. The enzyme activity assays measure, of course, the activity of the enzyme, whereas the ELISA assays measure the mass of the protein. In hematological assays, as we mentioned, they give very similar information, but in solid tumors, the ELISA methods are more sensitive, probably because they're also detecting the inactive forms of the TK1 protein.
Can tumor proliferation be identified at metastatic sites by this assay in the absence of tissue from the primary site?
Well, I can take this one, and yes, you can use it to study the proliferation in metastatic sites. That is, if you have removed the primary tumor, if the TK level is still high or maintaining at an increased level, this is a clear-cut sign of a metastatic process where there is cell proliferation going on at other sites. In order to localize it, of course, you need other methods like imaging.
All right. What are some cancer cell proliferation biomarkers in leukemia?
Well, that is the type of studies and use that is presently the major one for the activity assay. The TK Liaison, for instance, is used, and quite for many years and in several different clinics just for this purpose. So it's definitely there to be used mainly for monitoring treatment in leukemia and detect recurrent disease.
Okay, thank you. how does this biomarker compare with circulating tumor cells and cell-free tumor DNA?
We haven't looked particularly at that study. I think my attitude to biomarkers is all biomarkers are useful, so I'm sure that if you combine TK210 with these biomarkers, you find it's a 1 + 1 = 3 situation. But we haven't done any we haven't ourselves done any studies on this.
All right, perfect. What level of sensitivity and specificity do you think is required in order to use it as a marker for early prostate cancer?
Well, this is a tricky question, and of course, prostate cancer is a relatively slow-growing process, so we do have to have very high sensitivity and specificity, and there is only one way to know this, and this is to do the studies, and that's what we are doing presently.
Okay, perfect. And I do want to remind everyone that you will be presenting some data on early prostate cancer at AACR, so keep an eye out for that. I don't see any further questions, so I would like to thank our presenters, Stefan and Martin. Thank you so much for your presentations today. I'd like to thank the folks at AroCell for sponsoring today's web symposium. And most of all, I'd like to thank those of you who came and spent some time with us. I hope you found some information that will make your research a bit easier going forward. Thank you again for coming, and have a great day.